give that amino acid off during the process of translation. Okay, then when it gives it off, it's empty again. Now it can go and pick up, will it pick up the same one or will it pick up a different amino acid? Different, what? Well, it will be a new one, but it will be the exact same version, right? So if it left off a tryptophan, it's gonna pick up a tryptophan, right? Because it has not changed the anticodon at the bottom, so it's very specific to its amino acid it's gonna go and pick up. Yeah, they can't be the same one because that's now attached to the polypeptide chain, right? But it would be the exact same version. All right, so know the tRNA has the amino acid site and they have an anticodon at the bottom that corresponds to the codon of the mRNA strand. We're gonna look at this a little more, right? So we have a ribosome has two subunits. Tell me what they're called. The small and the large. You need to know what each of them are used for. The small one is used for attaching the mRNA. Very good. That has the site for the mRNA to attach to. The large subunit is used for, yes, making the polypeptide chain, making the actual chain of amino acids strung together. It has the A, P, and E sites, and that's where that happens. That's where those are strung together, the actual polypeptide. Don't memorize it in EPA, right? We're used to reading English from left to right. I want you to memorize it in the other direction, APE. Because E is the exit. Well, APE is easy to memorize, right? So that's nice. It's often illustrated in this way because, right, like we often illustrate these ribosomal subunits, but it's a three-dimensional thing. So we could look at it from the other direction, right? I want you to memorize it as APE. A, because that's where we're going to start. You'll see that's where the tRNA comes in originally, bound with an amino acid. It moves over to the P site. At the P site, it actually is where the amino acid is bound to the previous amino acid that was left off. At the E site, it exits. So you need to know what happens at each site, but that's all that you need. Simple as that, right? 8 comes in, binds to the small subunit, actually checks there, actually gets checked. The ribosome will check, okay, is it the correct base pairing? Is it the right tRNA? Does it have an amino acid? Okay, P site, okay, that's the site. This is the wrong, it's not named P for polypeptide chain, but that's how I memorize it. I think it's useful, right? P for that's where we're gonna grow the chain. And E, easy, because it's exit, right? That's where it's gonna exit the tRNA. Tell me, is there an amino acid at the E site stuck to the tRNA? No, right? Because it gave it up in the P site, right? The amino acid from the tRNA was left off at the P site, and now when it gets to the E site, it's empty, and then it will float off and can pick up that same amino acid, different version, or different molecule, but the same version. Okay, so A, P, and E, memorize those. Now we can get to the reading frame a little bit. So you read this in codons, so letters of 3. Each codon is complementary to the anticodon on a specific tRNA molecule. And to correctly read the mRNA message, translation must start at the correct location. What is the correct location? It's called a start codon. What do you think our start codon is? AUG. Okay, this is the only codon I'm going to tell you to memorize. All right? For the rest of it, we have what's called a codon chart. You might have seen this in high school. I hope you did, if you had any decent biology. Codon chart, you might be recognizing, they're not hard to read. Actually, most students do great on that on the exam. Okay, so they're not hard to read. You don't have to memorize the codons, but this one I do want you to memorize. It's called the start codon. It also, it corresponds to a met, okay? It makes, so every protein will actually start with that amino acid. You could chop it off if you want later in some modifications, but it starts with that always, okay? That is because it's very important to know where to start. What would get messed up? What if I'm in the ribosome, I start here, UGC. Every single amino acid will be wrong, right? Because I've switched everything now, right? So every one of them will be wrong in that case. And that can happen with mutations, right? If you enter a base pair in there, you can actually do a reading frame shift, right? All right, so it's important to start at the right location. This is the start codon, it's AUG. And that establishes what we call the reading frame, which is what I just said, right? The reading frame means what did I say? The fat cat sat, or whatever I said, right? The fat cat sat. It establishes that that's where we start. So we're gonna read in threes. We're not starting at the hut, fat, sluts, right? That becomes a whole new sentence. That's all that the reading frame means, so that the right groups of three letters are read as a codon, and the start codon is AUG. Right, so memorize that one. It's the only one you have to memorize. Now, translation. What steps did you have for transcription? I told you memorize 1, 2, 3. They're called. Yes, good. And then, yes, and very good. You have the exact same words for translation. Okay? So good, because, okay, it held true for transcription. It's going to be the same steps called for translation. Of course, in the slightly different things that happen, because what are you making here? You're making protein, you're not making mRNA, right? But kind of visually you could understand, right? Like, okay, they're called this. Initiation means that you find the start codon. It's located on the mRNA, and then the two ribosomal subunits come together. This actually, because what does it bind to, the mRNA? Which subunit of the ribosome? The small, okay? The small subunit has the mRNA binding site. So that happens, then it's actually like the bun comes on top. So the large subunit comes on top. That contains the APE sites, and then can add those can attach the amino acids together that the tRNA is bringing in. Then you have elongation, and for elongation this time, it's not the same as for transcription because we're not adding RNA nucleotides, we're adding the amino acids together, but we're still growing a chain of something, so we call it elongation. So that just means, okay, these are being made, right? A chain of amino acids is created in elongation. And then termination, okay? Actually here we have a stop codon. Do you see here, UAG? There's actually 3 of these stop codons. You don't have to memorize the stop codon sequence, but you should know what it is, right? Like, okay, a stop just means we're done, the protein's done. We can be done and let go. All right, and then of course the ribosome a new one comes in, right? Okay, a new mRNA comes in. Okay, so it releases the amino acid chain and boom, it's done. This is actually worth watching.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes
See this
each
red
amino
thread
acid
here?
and joins it onto the growing protein chain.
That's the protein chain, the polypeptide, right? The amino acids are
As the
coming out there.
mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
All right. So here's that table that I was referring to, the codon table. And you see some stuff here that I told you don't have to memorize. Do you see those in red up there? How do you think you read this? If you have this sequence, it starts with AUG, what would be, tell me, what would be the next one? Do it with a friend. CCA. What amino acids do we get out of CCA? Which one did we get? Proline. Okay, you don't need to know, right? It comes in 3 letters, right? And that's all you need to know. You don't need to tell me on an exam like, oh, that's proline, right? The abbreviation is just fine. I have a hard time actually with certain of these to pronounce them myself because English is my second language. It doesn't roll off naturally for me. But how would you do it? CCA, you would do it first, right? Just like other charts where you use this. The first base would be C here. The second one is also C, so then we get to this one, right? And then luckily all of these guys are proline, right? Do you see something funny here? Different codons encoding for the same amino acid, right? That's what this means. It's degenerate. It's all it means. It means that there's several codons for one amino acid. Now, I'm going to ask you a question. Do you ever see one codon encode for more than one amino acid? No. No. What I'm trying to say is that CCG, because could CCG become any other amino acid than proline? No, because that would be terrible, okay? The whole point is that you need to know exactly what amino acid is going to be in the protein, right? That's what your DNA code is for, is to determine the exact amino acid sequence in that protein, right? Because that determines what the protein is going to be, what it's going to do, what the shape is going to be, and what the function is going to be. And so, yes, several codons, one amino acid, but not the other way around, not several amino acids from the same codon. That would not be the case. And that's important. If you really think about it, you'll get it, right? Like, okay, I need to know exactly which amino acid will get in here, and I can have several codons that would give me that amino acid, but I could never have one codon give me more than one, right? That would not make any sense. Okay, now it actually is helpful to have this. Do you see which one of the bases is it that can change? Look at proline that we were looking at. Which one differs? Which of these letters? The last one. We call it the wobble base, okay? The last one can change. Would that be useful if you have a mutation? If you have a base pair substitution, which means that one base pair is substituted for another base pair, actually, if it's the last one, you might be fine because you might get the exact amino acid that you would get from the start, right? So let's say your code was supposed to read CCU, now it reads CCA. Doesn't matter. We call that a silent mutation. It would be silent because you would have the exact same amino acid and thus you would have the exact same protein, right? So it's actually good that we have this, right? It's a great thing that it is degenerate because we now have room for error, right? Okay, so the last letter in a codon can be changed without major consequences, and it's called the wobble base. The wobble base. Okay, you can also see that some of them, right, have more versions, right? Like proline has 4, but histidine only has 2, right? You don't have to memorize any of that, but that's the case, right? So histidine is something that it's, you know, you have a mutation, it might be something that's not silent. It might actually be switched out to a different amino acid because it has less wiggle room, less wobbling. Okay, so I told you to do the first one, right? So we're gonna skip this for now. I think you got it. Does anyone feel like, no, I don't understand how to do this at all? If you have this sequence, can you do the translation using the codon table? Yeah, if you can't, absolutely come and see me, right? Come and talk to me after class. I do have time. I can go to my office and do extra office hours, okay? Today, I'll have some time before lunch. So definitely come and see me. I'll help you with this. But literally, you read first letter, second letter, and you find that corresponding, okay, it gives me whatever. That will show up on the exam, right? But usually my students, I mean, I don't see a lot of people mess up with that, so. Okay, so basically what we've done is we've gone the whole way from the DNA plan to the actual protein in the end, and you can kind of look at it the same way as the cookbook example, right? We had the actual blueprint or grandma's cookbook, okay? We made that into a different instructions that could be read, right? So the mRNA can be read, or you put it on an index card for your friend, right? The cookbook, and then they're actually using, you know, structures, they're using bricks and what have you to actually make the building. And of course, we're gonna use amino acids to string them together to make the actual protein. Okay, this is a good one. So let's do this. For each of these processes, circle the things that it uses. So look at transcription. This is a good one for practicing for the exam. So transcription, which one of this does it use? Translation, which one of this does it use? So also, what nucleic acid do we start with? I've been saying this a million times, but that you have to have down what it ends with and where it happens. Okay, and then circle the ones that it uses. So do it with a friend, try to do it quickly, and then talk to your friend if you're unsure. And don't wait for me to give you the answers, because then that's not using the time wisely.
But like, how do you get to open? You download the PDF. You have to download the password. Right here. And then go back.
All right. So transcription, what do we start with? The nucleic acid. We start with DNA. Great. What does it end with? mRNA. Where does it happen? Nucleus. And what does it use? Does it use amino acids? No, right? Okay. Does it use mRNA? I would say it makes it, right? Like, so uses it? No, not really. It makes it, right? It produces it. Ribosome, so this is not a question, doesn't show up like that on the exam. I want you to, so I would say produces it here, right? As a note. Okay, ribosomes. Do we use it during transcription? No, right? We're inside of the nucleus, so no. Nucleotides? Yes, right? RNA is made out of nucleotides and so is DNA. Very good. What about tRNA? No, absolutely not. That is for translation, right? Brings it in at the ribosomes. What about RNA polymerase? Yes, right? Making DNA to become RNA needs RNA polymerase. When do we use DNA polymerase? When we're making DNA. In replication, right? So that's different. And I'm saying this because I do see people confusing the different processes, and it's good to compare them to understand. Okay, what about this guy? Translation. We start with mRNA. Specifically, we start with the mature mRNA, right? The actual processed mRNA. Okay, and what do we end with? A polypeptide chain or a protein. You can answer, those are use those terms interchangeably. It will help you if you understand. Okay, it's a polypeptide chain is the same thing as a protein. It just actually doesn't mean that it's been folded yet. Okay, where does it happen? Ribosome. If you put cytoplasm, fine. As long as you know that it's at, a ribosome is involved, it's at the ribosome in the cytoplasm, that's fine. Okay, and what do we use? Do we use amino acids? Yes. Do we use mRNA? Yes. Do we use ribosomes? Yes. Do we use nucleotides? No, we don't use them, right? We're reading them, but like not really. We're not using them to assemble the structure, right? So I would say for this one, yeah, reading them, right? Make a little extra note to yourself. Again, we would not use that terminology in an exam question. Do we use, right? Because that's too confusing. Yes, we read them. Okay, tRNA. Yes. RNA polymerase? No, right? We're not making any RNA. All right, now every cell in your entire body has the exact same genome. This means that all of your cells have the potential to make any of the proteins your genome encodes, okay? I've told you this 100 times. All of these cells are different, right? All of the cells are different, and so you need differential gene expression. This is what cells use to selectively express only the genes that encode proteins they actually need, okay? This is called differential, right? Because you are making non-specialized cells, we call them stem cells, to become specific types of cells, okay? That's all that you need to know about differential gene expression. Understand that, okay, we need to selectively express genes that encode proteins they actually need. They become specialized cells. And this is the thing about the transcription factors I was trying to emphasize before because they're so important for this, right? So So transcription factors are the proteins that help with the differential gene expression, right? So you have transcription factors, you have repressor proteins, they block the RNA polymerase, right? So what if you have that protein sitting there blocking it at the promoter? Well, then RNA polymerase can't bind there and can't make the mRNA, right? So then you won't get that protein in the end, because if you don't have the mRNA, you're not going to get the protein, right? So then you have activators that will increase the rate of transcription, and that increases the protein production, right? So you can decrease it for some cells, you can increase it for other cells, and there you go. Now you have different cells expressing different proteins based on this. Okay, so it doesn't say here, you know, promoters or the binding sites, but you should kind of know that from the previous slides, right? This is where that region is involved, and that would help you. You actually don't have to understand this figure, but I think it helps when you're reading, when you're studying. Just understand, okay, repressor proteins, they block that, they would block the RNA polymerase. Activators, actually, they kind of sequester RNA polymerase, like here, come here, right? Like, here we are, like, come over here, make more of this stuff. Okay, this is actually not super important. This is for, because we've been talking about insulin signaling and eating that donut in the beginning of the class, but it's kind of cool. For those of you that are interested, definitely read about it, right? But you should understand, if you have insulin resistance, right? These cells, they make insulin, but the body cells don't respond to it, right? But then, if you have insulin resistant, you actually will have, right, you can have a decrease in the expression of the receptor. And if you don't have that much of receptors, then boom, there you go, it can't sense it anymore, and you can't take up the glucose. So that's not hugely important that you understand this concept, but it is cool for those of you that are interested in diabetes, and you're definitely going to see it in your profession moving forward. Okay, mutations, last thing. What is a mutation? Changes to the DNA, right? Or changes in either, actually, replication is an error, or transcription and translation, right? Something went wrong in the processes of making the protein. So it could be one base pair being replaced to a different base pair, meaning a T now is an A or a C is a G, right? Shouldn't be there. Or it could be bigger chunks than that, okay? So it could be mutations. You have everything from a whole chromosome, right, entering the cell, like in trisomy 21, or little base pair changes. Do you have mutations right now, sitting here? Absolutely you do, right? Sometimes these mutations become advantageous for the species, in which case they increase, right? Sometimes they don't do anything, they're there, don't do much, hang around. Sometimes they're detrimental. Okay, so if you have a normal gene, you have a mutated gene, you can get an abnormal protein, no protein. I would actually make another arrow. You can actually, from a mutated gene, also get the normal protein. That's a silent mutation, right? Okay, if they're not repaired, the proteins may be misshapen, and they could potentially be non-functional. Sometimes they're actually like halfly functional, so you'll still have kind of a response in whatever pathway, but not the full normal response. Okay, point mutation, it is what I told you. One of the point mutations could be a base pair exchange, right? It's one base that's been changed. Either it's a deletion. What would a deletion be? The whole thing is cut out, right? But it's only one base pair. Or an insertion, meaning one thing is inserted in, right? Or it's a base pair substitution, where you have switched one for the other. Tell me which one of this would be least detrimental. Talk to your friend for a sec. I hear it. Very good. A substitution. I hear. Very good. Okay, why? Because you still have the, what was the term we called it though? Cat, hat, set. You still have the code, you still have the reading frame. Very good. You still have the same reading frame. And so maybe you've changed one amino acid, but all the others are going to be there, right? So, yes, an insertion of one nucleotide. That's not good. That changes the reading frame. A deletion of one nucleotide, that's not good. That changes the reading frame, right? All of those are actually called point mutations. They're under that umbrella term of point mutations, but some of them have much higher, like they don't, yeah, it sounds like point mutation is not that bad, but well, if it's a reading frame mutation, that is certainly not good, right? So this is what's called a frame shift mutation, right? Reading frame of the mRNA molecule will shift. Okay, this base pair substitution, right? You can have what I called a silent mutation, right? We had that, proline became another proline, remember that? So that means that the nucleotide has changed, but it does not impact the amino acid added to the protein. Then we have the opposite of that. It's just called a missense mutation, and that just means that, okay, it has been changed. So now it was supposed to be a proline, and it's now a leucine or a tryptophan, right? It's not the same amino acid anymore. We changed the base pair out for a different one, and now it actually has an effect. That is called a missense mutation. Then we have another one that you need to know, and it's kind of my favorite. I love the person who named it a nonsense mutation. Why is it called a nonsense? It made a stop codon. Okay? Imagine that you put a stop codon as the second. So here, I put a stop codon. Accidentally, I made this into a stop codon by a mutation. Are you going to make the rest of this? No. So there you go, very, very dangerous mutation to have early on because you're not going to make the rest of the protein, right? If it's at the very end and you're just missing one amino acid, maybe it's not that bad. But if it's early on, that would be really bad. Okay, so when you're studying at home, look at the frame shift mutations, right? Look at this reading sequence, frame shift mutations, right? Shifting everything. The mRNA nucleotides will be read in incorrect groups. There's even better examples of this where it's actual words like cat, sat, hat, right? Like where you can clearly see that the whole sentence will be disrupted in this. So this is, it says +1. We've added one nucleotide, right, into the thing. This says -1 because it's deleting one nucleotide, just like I was trying to tell you, right? What would be worse, this or a base exchange, a base pair, right? These two are worse than just doing a base pair exchange. Okay, so you should know that some of these terms actually go together, right? So if you have a missense mutation, that could also, a missense mutation could create a nonsense mutation, right? So I know some students want to say like, oh, this is a missense mutation and this is a stop, but understand missense just mean new amino acid, right? Like a different one. And that in turn can be also grouped into a different type of mutation. So don't try to be so strict that you're putting one mutation into a certain category. Sometimes there's two things going on, right? Okay, so nonsense, we said a new stop codon is generated and that would stop the whole thing. Oh, here it is. Oh, good, I had it still. Okay, I was hoping for this. Right, so, no, it's not this one. It's the next one. Okay, good. All right, reading frame, right, 3 nucleotides. There's a question like this on the exam, I think. What's worse, adding 1 nucleotide or 3? Well, 1 is worse. Because if you add 3, you've just added 1 amino acid, right? And imagine you have hundreds of amino acids in a protein. Adding 1 might not be that bad, wanting 1 little amino acid. But if you add 1 nucleotide, what do you get? What kind of mutation do we call it? A reading frame, right? A frame shift mutation, which means everything is going to be wrong from then on. There it is, thank you. Okay, there it is, right? So if you're looking at this, the cat ate the rat, right? I don't like this example exactly. It should say cat with a C, if you ask me, right? The same exact amino acid is what you get when you do this one in the silent mutation. I don't love that they put a K there, but whatever, you get the point, right? Silent mutation, it stays the same, right? In a missense mutation, the sentence will be changed. A deletion, okay, specifically, right? 1 or 2 base pairs, the whole thing gets shifted, so now it's, right, this is the original sentence, the kateterat, right? That doesn't make any sense anymore. Or you insert 1 plus 1 and you have the ekatat et erat. Yeah. Okay, so I'm gonna leave, I think we're gonna leave here. We're out of time. So look at this slide for yourself. You already know some of these carcinogens, right? These things that can cause mutations. Understand that they can be external, like the sun, cigarette smoke, pollution, or eating bad foods that have carcinogens in them. But they also can be internal, right? So you just making mistakes. Do you think you make more mistakes as you get older? Yes, okay. So I'm getting to that age where cancer is increasing at a rapid rate, right? So this is why cancer goes up with age dramatically. The older you get, the more likely you are of getting cancer, because your processes of replication, transcription, and translation are not as good as they used to be. Okay, there we go.